Hi, I'm Pamela Wenzel, Professor of Integrative Biology and Pharmacology at UT Health Houston. Today, I'll tell you about our article published in the Journal of Experimental Medicine entitled Mechanometabolism Instructs Hematopoietic Stem Cell Specification. As background, a healthy blood system ensures that we can fight off disease, deliver oxygen and nutrients to the cells of our body, and recover from injuries. When the blood system doesn't function properly, one of the many possible treatments is a hematopoietic cell transplant. Transplantation of hematopoietic stem cells can be curative, but many patients don't have a suitable donor, and for those that do have a donor, the treatment carries risks of infection and graft versus host disease. In our article, we aim to understand how the environment impacts specification of the first hematopoietic stem cells produced in the body, with the goal of engineering stem cells with improved blood repopulating activity. During embryogenesis, the first hematopoietic stem cells emerge from the walls of arterial vasculature within 2 to 3 days of initiation of the heartbeat. And here, during a highly restricted time frame, they transition from an endothelial to hematopoietic identity as they bud into the luminal space and enter the bloodstream. While attached to the vessel wall, cells are exposed to stretching, pressure, and shear stress. Wall shear stress is the frictional force a distance of 0 from the vessel wall. And in prior studies, we showed that wall shear stress from blood flow promotes blood development and specifically the generation of hematopoietic stem cells by elevating levels of nitric oxide and prostaglandin E2. Please check out our new article where we show that wall shear stress also activates the mTOR signaling pathway as a critical part of the process that specifies hematopoietic stem cells. We find that this force-responsive signaling increases translation of mRNA transcripts containing a specific nucleotide sequence at the 5' end of the mRNA. The selective protein translation drives remodeling of the mitochondria, which is characterized by changes in the ultrastructure and proteins in the inner mitochondrial membrane. So interestingly, cellular changes like these would typically negatively impact adult hematopoietic stem cells. But for specification of new hematopoietic stem cells in the embryo, this signaling is essential. I'll tell you about 3 of the several important observations we made. The first important finding was that only cells with high mitochondrial membrane potential are able to reconstitute the blood system of adult recipient mice. Additionally, cells with high membrane potential are better equipped to generate T cells of the lymphoid lineage. This supports the idea that cells must build mitochondria to acquire hematopoietic activity. The second important finding was that activation of mTOR signaling by wall shear stress appears to occur selectively in hemogenic endothelial cells before acquisition of hematopoietic identity. Additionally, we found that only smooth laminar flow could activate mTOR, and disturbed flow had little effect. The third important finding was that we were able to use a drug that activates mTOR to improve the ability of hematopoietic precursors in the embryo to reconstitute the blood system of the adult when transplanted. Together, these data support our hypothesis that wall shear stress contributes to the expansion of hematopoietic stem cells by stimulating mTOR-dependent metabolic reprogramming. And we believe this new knowledge could be leveraged to improve blood repopulating activity of hematopoietic stem cells and as a powerful tool to continue studying blood diseases. So please check out our new paper where you can find more about this study and how we also found that wall shear stress alters the mitochondrial proteome and ultrastructure using culture systems of pluripotent stem cells and embryos without a heartbeat.